Hello, everybody. This is going to be our first lecture on the topic of molecular biology, molecular processes like DNA replication, transcription, and translation. We're going to do it all. Okay, get ready. And so on this first lecture, we'll be covering some of the content in chapter 10, parts 1 through 5, and then we'll move on from there. Okay. So let me start by going through some of the context here. In fact, we always like to begin with a discussion of the history. This is kind of like the Mendel discussion that we started off our discussion of genetics in. Chapter 10, part 1, covers some of the experiments like Fred Griffith's experiment, very briefly, but we move very quickly into this experiment by Alfred Hershey and Martha Chase, whose work is just so universally known that people refer to the Hershey and Chase experiment, and everybody knows exactly what you're talking about. Okay, and so I want you to be in part of that informed crowd too. And so what you need to be aware of here, well, there are a few things, a few main points here. One is that we're involving the bacterium, E. coli. It should be underlined or italicized. And E. coli is a bacterial cell. And the phage, the T2 phage, is a virus. Right? So the E. coli cell is the host of the virus. And basically the virus, the phage, is using E. coli as the mechanism that it uses to make lots more copies of the virus. Okay, and so yeah, even bacteria have viruses. That's a pretty spectacular thing for some people to realize. Okay, the other thing is that we're looking at a time in which we weren't really sure about, well, what kinds of molecules were important for heredity. This was before Watson and Crick. This was in the time, we knew that genes were important, right? We had the concept of genes since the rediscovery of Mendel's work. And so people were doing experiments with fruit flies, with mice, with corn. But even at that point, we really didn't know what these genes were. What were they, right? What were they physically? We knew that there had to be some kind of molecule inside of cells that was capable of transmitting information, the hereditary stuff, right? But what was it? The two candidate models that we were thinking of, the strongest candidate was proteins, because we knew that proteins were extremely diverse and very, very complicated in their structure. You know that. You know that we've got 20 different amino acids. They're there in particular sequences. That much we kind of had a pretty good idea of at the time. And so it seemed like proteins, being as complicated as they are, would have been a great candidate to be the stuff of heredity. The other candidate was DNA, or nucleic acids generally. We knew that DNA was there in abundance in the nucleus. That much we knew. It didn't seem like it was that great of a candidate because it looked like it was just kind of structural. It was occupying space and had this relatively inert tendency. It didn't really interact with too many other kinds of molecules. It turns out that the fact that it's inert and not terribly reactive is kind of what makes DNA a really good molecule to be the hereditary stuff. Right? It's not going to change because it's reacting with lots of things. And so, but at the time, it seemed like DNA was just kind of a minor player. We're almost sure that proteins had to be the stuff of heredity. Okay? And so what the Hershey and Chase experiment did was it basically said, okay, well, I'm going to give you a really good experimental design that will help me make specific predictions about what to expect if proteins are the stuff of heredity versus what to expect if DNA is the stuff of heredity. Right? And so that's the context of what Hershey and Chase set out to do. The other thing that you need to remember here is that proteins, because of what it is, right? Yeah, it's made of amino acids, and sometimes it has sulfur. Right? Sulfur is one of the elements that's there in the body, and it's primarily there in proteins because there's one amino acid, a pretty common amino acid, cysteine, which has sulfur as a component. Right? There's a sulfur atom in every cysteine amino acid. And so sulfur is a definite presence in proteins, but it's not present in DNA. And DNA, on the other hand, has phosphorus. And we've already talked about this. You know this because you know that one of the bits of a mononucleotide is a phosphate group, which is PO4, right? P is phosphorus. And so we could use this compositional difference between nucleic acids and proteins to make this decision. Is the hereditary stuff proteins or is it DNA? And this is also the time when we're learning about radioactivity. And so it's possible to radioactively label phosphorus or radioactively label sulfur. You can grow viruses and bacteria with media in which all the sulfur is radioactively labeled. And if you do that, then the phage that come from that you're able to isolate the phage, is going to carry radioactive sulfur. So it'll be glowing, right? Or alternatively, you could grow the bacteria and the viruses in a medium that has radioactively labeled phosphorus, in which case the DNA and the RNA, but not the proteins, are going to carry this radioactive label, right? And so because we're putting a special marker on the phosphorus or a special marker, radioactivity marker on the sulfur, we're able to make these really clever distinctions. It's a super clever experiment. And I want to go to the textbook right now to show you what's going on, right? And so I know you've been to the textbook before, and so you can just speed through this relatively quickly. And this is a picture of what a bacteriophage looks like. It's actually different in structure from the viruses that infect us. I mean, some parts are similar, right? There are a lot of viruses that infect animals that have an icosahedron, this regular shaped head. It's in one of our viruses, we would call it the capsid, and that's one of the shapes. But the head of these viruses, the shell and the tail fibers, all the hard parts, the outside of the virus are made of protein. And so when you use radioactively labeled sulfur to make bacteria and viruses, and you collect the viruses, all the protein, all the shell is going to carry radioactivity. The DNA, which is on the inside of the viral head, right, is going to not be radioactive. It's just going to be regular DNA because the DNA has no sulfur, and there's nothing in the DNA that will cause it to carry the radioactivity, right? On the other hand, if you were to grow the bacteria and the phage in a medium that contains radioactively labeled phosphorus, then the shell and the tail fiber is not going to have any radioactivity. All the radioactivity will be in the DNA, right? And so that's what they do. They have got the hot sulfur and the hot phosphorus, and they apply it to the bacteria. You put these radioactively labeled phage into with clean bacteria, okay? And then once the bacteria become infected, then you could separate the phage shells from the infected cells. And what do you find? Well, the proteins they stay in the liquid. And when you centrifuge it, the pellet contains only the bacterial cells. And so what happens is that if the radioactivity is in the protein, the radioactivity stays in the liquid portion. It doesn't go down to the pellet. And that tells us that, whatever the protein component of the phage is not what's going into the cells. yeah, at that point, the question is, well, are we sure it's the DNA? Well, that's where the other half of the question, the other half of the experiment comes in. Because if you were to predict the DNA is what gets put into the cells that are infected, then you would predict that when you do the same experiment using phosphorus that's been radioactively labeled in the phage, then the radioactivity will go into the cells, and that's exactly what happens, right? We're actually seeing the radioactivity in the pellet, and that's telling us that it's probably the DNA, right? And yeah, I know that's a lot of explanation, and you probably were hoping that I would just kind of gloss over this, but no, this is really neat stuff. I mean, it's historically important because this is one of the foundational experiments that let us know that it was indeed the DNA. So, all right. So yeah, it's only from that point that we get further confirmation, additional studies using different systems, and eventually that leads up to the structure of DNA, which is going to bring in our players Watson and Crick together with Rosalind Franklin, a major player. And so the next thing we're going to do is to review the structure of DNA, and you know the story. We did this way back in Unit 1, right? You know that a mononucleotide has a 5-carbon sugar, right? Here are the 5 carbons: carbon number 1, carbon number 2, carbon number 3, carbon number 4. Oh, wait, I didn't draw that right. Let me undo that. Okay, so I think I drew a 6-carbon sugar. Okay. So let me try that again. Okay, so this is going to be, this will be carbon number 1, carbon number 2, carbon number 3, carbon number 4, and then carbon number 5 is going to be up here. And so this is going to be either ribose or deoxyribose. Right? We know that from the number 1 carbon, we get the nitrogenous base. It's going to be A, adenine, C, cytosine, T, thymine, or G, guanine in the case of DNA, and in RNA, uracil replaces thymine. And then we know that on the number 5 carbon, we have a phosphate group, okay, which in the case of a mononucleotide triphosphate is going to be carrying 2 additional phosphates. And remember that these are the extra phosphates that are going to create the unstable bond right there. Mononucleotide triphosphates is a starting point from DNA synthesis. And so because we're putting on this molecule, it's going to be this is going to be a significant point for us later on in the discussion. Right? We talked about this. We said that DNA and RNA mononucleotides come pre-charged with their own energy. And that extra phosphate group, pyrophosphate group, is the thing that makes these things pre-charged. Okay. We also know that the growth of the molecule is going to happen when we add a phosphate group. Okay, so that's going to be the new bond that forms. Let me go back to blue. Phosphate group of the next that's a funny looking ribose is going to be our next nitrogenous base. But basically what's going on here is that we make these bonds by breaking off the phosphate groups. This bond right here is broken, and we're able to create a new bond between this phosphate group and the number 3 carbon off of the earlier molecule, right? And so what I'm getting at here, and this is not something that is stressed in this portion of the book, although it probably will become later very important, is that nucleic acids are always going to be adding on to the number 3 carbon. The next mononucleotide triphosphate is going to be coming in over here, and the next one's going to be added on to the number 3 carbon over there, because at the other end, at the far left-hand end, oops, at the far left-hand end of the molecule, we don't have phosphate group. We just have a single phosphate. And so we're going to be adding on to the number 3 carbons. And so we say that at one end of the molecule, we've got a 5' end. That little apostrophe is, we say 5', okay? Recognize that. Into this other end, we would say this is a 3' end. And so we're going to be saying, well, the molecules of DNA and RNA are always going to grow from the 3' end and not from the 5' end. Okay. Okay, there's that. Okay, the other thing is that if you have 2 molecules of DNA, so if you have the 2 strands of DNA, I'll do one in blue, the other one in red, right? One strand is going to be going in one direction, 5' on one side and 3' on the other end, and the opposite strand is going to be running anti-parallel. So 5' on this side, 3' on this side. Okay. And this is another characteristic of DNA. The 2 strands are anti-parallel. And these are things that actually are not terribly well illustrated in the textbook, which is why I'm not relying on it, although we'll be going back to the textbook pretty soon. Okay? And ultimately what we're getting at with today's discussion is a talk about, well, the process of DNA replication. Oops, just gave you the answer here. Right? Let me back up a ways. So do you remember what happens during the cell cycle, during that middle portion of interphase? Remember, between mitoses, we have a long interphase that goes G1, S, and then G2. What happens during the S portion of interphase? Well, yeah, of course, I just told you the answer. DNA replication. We start off with one long strand of DNA, we end up with 2 identical copies. Right? How does that take place? Well, that's what we're going to be talking about today. Right? And so we're going to be saying, well, DNA polymerase operates at replication forks. DNA polymerase requires a 3' end to build off of. You know that's the case. And as a consequence of this constraint that DNA has to grow in a 5' to 3' direction, one of the strands at the replication fork, one of the new strands is going to be synthesized continuously, while the other one is going to be synthesized discontinuously. And let's see what the textbook has for us here. Okay? Because, yeah, I'm actually going to tell you ahead of time that it is pretty inadequate what the textbook is giving you here, because we really are going to be needing to think about that 5' to 3' directionality in the laboratory, particularly in the PCR laboratory, where we have these 2 things that are called primers. And why do we need primers? Well, to understand that, you really have to understand that we've got this constraint. DNA cannot grow in the 5' end. It only can grow in the 3' end. Okay, so here we were on the previous time we were at the textbook. Let's look at the next portion, the next chapter. This should look totally familiar. Oops, let me get my annotation scheme going. Here is our ribose, so deoxyribose sugar. Here's the nitrogenous base. Here is the phosphate group. And so this is our sugar, phosphate, base, phosphate. And this is one way of representing a polynucleotide, right? You can represent it like this, where you've got the sugar, the phosphate, and the base. And there are just so many different ways of expressing the same idea. And when we draw it this way, you could sort of wrap that around and create a double helix. We've got the sugar, the phosphate, and the base. Matching up with the sugar, a phosphate and a base, and you end up getting a nice long strand, which can then form a double helix with another molecule. Here are my drawings. Back to my mouse. There's a little video. Not too interesting. Yeah, ribose is a little bit different. Okay. Yeah, I mean, all this stuff is a really nice review of some of the things we talked about back in Unit 1 when it came to the structure of nucleic acids. Okay, more unfairness. Way back in the day, around the time that the structure of DNA was discovered, you had Francis Crick, James Watson working in Maurice Wilkins' laboratory. No, actually, they were working in their own laboratory. Maurice Wilkins was the head dude, and one of his colleagues, Rosalind Franklin, was responsible for taking pictures, the images, and basically, if you're a good X-ray crystallographer, you can look at the structure. They can look at this picture right here, and see from that you actually have a double helix structure, right? Franklin basically took the picture that led Watson and Crick to create this model of DNA based on a double helix, and Rosalind Franklin got no credit for her work. Wilkins, Watson, and Crick got the Nobel Prize, right? Sad but true. And so there's all that. Apart from that, really talking about things that we already discussed. Yeah, technically, Rosalind Franklin died, partly because she didn't know about the health risks of exposure to high amounts of X-ray, and she was working in the X-ray laboratory for many hours. And basically, if you die, you're no longer eligible to collect a Nobel Prize. So that's the excuse the Nobel Committee gave for not awarding Franklin with a Nobel Prize. Okay. Now, this is what we're talking about with, this is kind of another central concept, which is the idea that we'll, at a replication fork, right? The replication fork. This is the old. And so here's where we've got the unwinding, right? Here's where the old strands are unwinding from each other. So this old strand is going like this, it's separating from the other old strand, and then a brand new daughter strand is made to pair up with the old strand. And so if you have old Watson over here and old Crick over here, they basically separate, and we make a new Crick opposite of old Watson and a new Watson opposite of old Crick. Okay. And that's the concept of semi-conservative replication. It's a good lesson to have. Yeah, so definitely read the chapter on that. And here's where we actually get at the process of DNA replication. Okay, so remember that in the S part of interphase, during the S part of interphase, we've got a ton of DNA to replicate. And it's not like you can start at one end and work your way all the way to the other end. You basically have to start in multiple sites. And so the model that we're working at here is that you've got these origin of replication origins. And so right here, we're going to create a little blip, and one strand is going to do this, and the other strand is going to do that. And then as that blip opens up, we'll end up with a replication fork over here, and another replication fork over here. And these replication forks will move out, continually unwinding the DNA until they finally meet up with the other replication loops. And we end up with two complete daughter molecules. They're two complete molecules, double-stranded molecules of DNA in the process. Okay. Now let me go back to the other board, the whiteboard share. Oops. Okay. And we said that, well, DNA replication operates at replication forks. Do another picture here. So I'm going to use red and blue again. So here's my original molecule of DNA. Let's say right here we've got replication origin. Color. Replication origin is going to be right there. Okay. Now what's actually happening is that we're going to be opening up the DNA there and putting a primer. We're going to be creating a little primer that will come in and form a little space for DNA replication to initiate. Okay, so let me open that up again. So here's my DNA strand, but I'm going to draw that replication origin a little more openly. I'm not going to, I'll create the replication forks, but I want you to see that there is a primer opposite of the replication origin on one side and opposite of the replication origin on the other side, right? Now that little piece of green DNA is going to be 5' on one end and 3' on the other end, right? So maybe that one will be over here. This one over here is going to look like this. It's going to be 5' on this side and 3' on this other side. Okay? And what this means is that, remember that DNA replication is only going to be going off of the 3' end. We'll be able to add nucleotides on to this side, elongating, making a molecule that will be continually growing and growing and growing and growing and growing and growing towards the replication fork from this side over here, right? But we can't grow on this side because we don't have a 3' end, right? If we look at the other side, the bottom side, opposite of the red original strand, the 3' end is on the left. And so we could add new nucleotides on this side, have growth in that direction. And so the new strand of DNA is going to be growing in that direction, right? And what that means is that we can't synthesize DNA on this side until we've opened up enough of the replication fork, enough of the replication loop. So let's say we've got this here. Okay. Here's my original primer. We know that we've got new DNA growing off in the direction towards left here, but we don't have any synthesis on the right-hand side, right? We know that we've got synthesis going this direction. Just keeps on going. As more DNA is opening up on the right at this replication fork, we just add more nucleotides on to that strand growing opposite of the blue strand, right? What has to happen is that we have to open up far enough so that we can get another primer in here, right? If we open up another spot where a primer can come in there, then we could get a new, the next strand of DNA, I'll throw it in a slightly different color, growing in this direction, so that we're able to eventually grow out and match up and meet the original strand. So basically, what we say is that this strand, we have continuous growth, whereas on this strand, it's going to be discontinuous, because as this continues to unfold, So as we continue to have more DNA unraveling, we might have opened up a piece right here where DNA synthesis could be taking place until it eventually meets up with the original primer. We still have a 5' end on the right-hand side here, so we can't have any growth until we've opened up enough to allow for another primer to come in, right? In the meantime, the continuous strand will basically continue to make more DNA as that replication fork opens up, right? And this is, this is maybe more than you need for a BIO100 class, but it does kind of reinforce this particularly interesting aspect of DNA synthesis, which is that we have to go in a 5' to 3' direction, right? Now, the 5' to 3' direction is also important for us because when we do our PCR experiment, we only want to synthesize DNA for the gene that we're interested in. It's going to be the gene for tasting or not tasting PTC. So if this is a gene that we're interested in copying, only from here to here, right? What we have to do is we have to have primers that are going to match up the two strands after we separate them. So the first thing that we do is we heat this up, which will cause two strands to separate out. Two strands separate, we call that melting. And then we add the two primers, and the DNA primer is going to be here. One is going to be matching up with the very end of of one strand. It's going to be leaving a 3' end over here, which will allow DNA synthesis to progress down like this. Okay. So we end up with this strand. We also have another primer, different primer that's going to be matching up with the other strand. Again, on its ends, we have a 3' end over here. And so we could basically make double-stranded DNAs simply because we have these, we're feeding the system these RNA primers, the DNA primers to match up with the very ends of the molecule that we're going to be trying to replicate, right? So these two molecules are going to turn back into our original molecules of DNA with a red strand and a blue strand. Okay, I'm doing it more quickly because it's kind of messy. And so after we let things cool, we have our two strands back, and then we repeat the process. We basically come back here, repeat the process, heat it up. The primers are going to attach to the very ends. We make the new strands, and then we cool it down. And then we heat it up, have the primers attach, we make new DNA, we cool it down. And so we go through 30 cycles of this, and we end up with a huge load of DNA copies of this DNA that we're interested in doing some electrophoresis on, right? And so the reason why this works because we're giving it a primer, okay? Not primer, it's a primer that is specifically designed so it's going to match up with the DNA. It's going to be complementary to the DNA at this end, the 3' end of the DNA. This is going to be our existing DNA molecule that we want to copy. And so this might be CGAT, And so the primer that we're giving it will be running 5' to 3' GCTA, and that's going to give us a 3' end that we could add more and more and more and more bases to the process of DNA replication. That's what the DNA polymerase is going to be doing, right? The primers are kind of like a big part of the trick. The primer works because of the 5' to 3' directionality of DNA synthesis. Okay? So now we've copied, we've done everything. We've talked about this. DNA polymerase is the enzyme that does it. We have to have a 3' end to build off of. At DNA, at the replication fork, one strand is copied continuously. That's the strand that is copying towards the DNA replication fork. The other strand is copied discontinuously in short fragments. Okay. And this is particularly of interest to us because of its significance for that portion of our PTC genetics experiment, where we're looking at the different kinds of the different phenotypes, taster or non-taster. Right? Now, last thing, I'm just going to briefly mention this, that while we're in the process of making brand new copies of a huge quantity of DNA, mistakes will be made, right? And so as you're thinking about your genetics project and the whole phenomenon of mutation and how mutation has to happen in order for us to get these conditions, and the fact that we're not all affected by these conditions is telling us that our DNA replication is pretty foolproof. We don't make very many mistakes. When we do make a mistake, it can have pretty serious consequences, but in fact, we actually managed to do this without making too many mistakes at all. And that, I think, goes pretty much without saying that there has to be a pretty sophisticated mechanism to prevent mistakes during DNA replication. And if mistakes do occur, we would need to be coming in and repairing it right away, right? Okay, now I'm bringing this out because I think some of you may be working on a project on a genetic condition in which the DNA repair mechanism is the actual thing that is not working terribly well. Right? This is one of the ways that things can go badly, and you could end up with genetically conferred disease, especially that's going to happen. I mean, you could have lots of mutations taking place if you start disabling the DNA repair mechanism. That makes sense, right? Okay, not everybody's going to have that, right? And maybe nobody's going to have that, but it's one of the things that can go wrong. And because DNA repair is so important, I'm hoping that you can see that this is totally going to be messed up if you lose your DNA repair mechanism. All right, that's all for now.